Hello everyone, I am Alison. I work at the MRC Biostatistics Unit at the University of Cambridge. Welcome to our virtual science event for Cambridge Science Festival 2021. Today you will be hearing from one of our expert statisticians, Professor Thomas Jaki, and you're going to be hearing all about clinical trials and how these are used during a pandemic. The live stream will include a few quizzes and also a game. If you haven't already, can you please click on the Q&A icon? So that is a speech bubble icon with a question mark in it, and hopefully should appear in the top right of your screen. So if you can please open this, and then we will be using this to share the links for the quizzes and for the game. And when Thomas mentions at various points in his talk, please keep an eye on the featured tab in the Q&A, and the links will appear there at the times that Thomas indicates. We will also use the Q&A for your questions. Please do feel free to ask questions throughout the talk. Or if you would rather, you can wait till the end, but we will be keeping an eye on the Q&A throughout, so please do feel free to post your questions in the Q&A throughout the session. So if you can just bear with us one moment, and I will hand over to Thomas.
Thank you, Alison, for the introduction. I'm glad that you decided to join and were keen to find out how we identify potentially useful treatments during a pandemic. What I'm going to do, however, in the first place is I'm going to start thinking about good practices outside of a pandemic, and in particular, I'm going to think about a really debilitating disease. It's called sudden onset arithmophobia, or short SOA, which is a condition that's affecting students that are preparing for A-level mathematics examination. And the way that this disease manifests itself is that a student wakes up one morning and finds themselves utterly unable to solve even the simplest mathematical problem. It can last a few days, it can be persistent, lasting for months or even years, and unfortunately there is no known cure for it at this point. I'm saying at this point because Professor James Moriarty has developed a 2-hour hypnosis-based intervention, and he claims that it will cure 75% of SOA sufferers. And so, my first question for you is, would you undergo Professor Moriarty's hypnosis therapy to treat SOA if you are a sufferer of that debilitating disease? As Alison has highlighted, the link is going to appear in the featured section, so if you could go there and submit your response. I can see some responses coming in, but I'll give you a bit more time. Okay, so at this point it looks like about 3/4 of participants have chosen or would choose to undergo this new hypnosis therapy, while the other quarter has decided that this is probably not for them. Now, myself being a bit critical, I guess my concern would be that this hypnosis therapy might turn me into a toad. And so thinking about the claim that is made and linking that to one of the Sherlock Holmes book, I'm reminded of the question by Watson, what is the meaning of all this, Mr. Holmes? And the response being, I have no data, I cannot tell. And so the first question, that I've just posed to you, obviously, I didn't give you a whole lot of information to go with. All I've done is I've made the claim that the treatment is effective. It will heal 75% of sufferers, but we don't know whether this is completely made up or whether this is indeed based on some solid evidence. And I'm assuming that on the basis of your answers were based on either assuming this was done on a basis of proper evidence versus versus it being made on the basis of just a simple claim without any evidence to back it up. And so our question in this context should therefore be, so what kind of evidence would we like in order to be convinced that we should use this hypnosis-based therapy to treat SOA? Now, I guess from my background and from the title of the talk, I think it doesn't come as a surprise that I'm thinking what we should do is conduct a good experiment, collect data, collect evidence to compare, to establish whether or not this treatment indeed yields good enough cure rates, whether it's 75%, 80%, 60% is probably less crucial than in the first place establishing that it does bring benefit on top of not undertaking this treatment. Which reminds me of a quote from a very famous statistician, well, a while ago now, in 1938, where he's said to have said, to consult the statistician after an experiment is finished is often merely to ask him to conduct a post-mortem examination. He can perhaps say what the experiment died of. Now, the way that I interpret this quote is to highlight the importance of collecting the information, the evidence in a very meaningful way. And so, therefore, what we're going to spend some time on now is just thinking about good practices in setting up an experiment, setting up evidence collection in order for us to make a decision about whether or not this treatment is potentially useful for SOA sufferers. Now, one of the first questions that we're going to have to think about is what strength of evidence do we require? Effectively, no matter what the data look like, no matter what the evidence that we've collected looks like, somehow we're going to have to establish whether or not the evidence is in favour of our treatment or whether it is against our treatment or saying there is no added benefit from the treatment. And so the first question that we need to ask ourselves is, what's the level, what's the bar that we set to claim that, yes, I believe that this treatment is actually better than not undertaking this treatment. And this is not specific to experiment. This is a very general concept for any decisions that we make, whether or not we take an action or we do not take an action. But fundamental to this decision about whether or not we are in favor or against the action is the worry and the risk of making mistakes. Because as decision makers, what we have to do is we're going to have to choose between we take an action, and the action in our example would be we support the treatment proposed by Professor Moriarty, or we do nothing, meaning we stick with the status quo. Doing so, we're going to be exposed to two different possible errors. The first one is called a type 1 error, which is to take an action when nothing needs doing. And so, again, in the context of our SOA example, this means that we do decide the intervention, the hypnosis-based therapy, is helpful when really it doesn't seem to add anything. And in terms of setting up the experiment, by controlling, by setting what level, what risk of a type 1 error I'm willing to take, this can be used to define the height of my bar. If I'm willing to make this type 1 error often, then my bar will be relatively low, because it will be relatively easy to jump over that bar, but at the same time I will be jumping over that bar too often, or sometimes when I shouldn't have jumped over my bar. The second type of error, which I guess not very imaginatively is called the type 2 error, is to do nothing when we should have taken an action. And so in this case, in our SOA example, we could think about having a treatment that actually helps SOA sufferers, but we decided that no, the treatment actually, the intervention is not useful. And we can use the risk that we allow for a type 2 error to determine the amount of information that we require in our experiment. You can think of that as how many experimental units, how many sufferers should we include in our experiment. And by choosing how high of a level of a type 2 error we are willing to take, we can set the size of our experiment. So let's move a little bit into the setting of a pandemic for a short while and go back about, well, almost exactly a year, when there was a story in The Guardian that described that there was a shortage of protective gear, protective eye protection for NHS staff, and it was uncovered that the Department of Health had dismissed a call for stockpiling eye protection a couple of years earlier, on the basis of deciding that the cost and the effort to do so, to stockpile eye protection, was not outweighed by the risk that there would be a need for this eye protection in the near future. Now, obviously, one of the great things that we can work with now is hindsight, right? Because we know now that actually it probably would have been useful to have a stockpile of eye protective gear available at the beginning of our current pandemic. And so, with this in mind, my second question is, so failing to stockpile eye protection before the pandemic, was this a correct decision, a type 1 error, or a type 2 error? And again, you can use the link in the featured section, which will get you to the poll. I'll give you a bit of time again to think about the problem and respond. So just to recap, there was a suggestion in 2017 that there should be stockpiling of eye protective gear be done in the case of need later on. And the decision by the Department of Health at this time was the effort, the cost, and whatever else did not warrant this stockpiling. And the question is this a type 1 error or is this a type 2 error? And I can see the questions, the answers coming in, and the majority has chosen correctly type 2 error. So with the benefit of hindsight, it would have been great to have the eye protective gear available at the onset of the pandemic. The decision was not to do so, and therefore we would argue that there has been a type 2 error being made in this instance. Now that we've thought a little bit about how we can make decisions and the sort of factors that feed into the decision making, I guess we're going to have to think about what we ought to base our decision on. So far, all I've done is talk sort of conceptually about what kind of decisions we could make, that there is a bar that we want to cross over or not, that we can make mistakes and so on and so forth, but we haven't actually spoken about what the data would look like, what kind of information we would like to collect in order to make a decision about the utility of SOA, for example. And generally speaking, the basis for the decision should be, first of all, should be meaningful to the patients. It should be measured reliably, and by that I mean I should get the same answer every time I measure. Think about using a weighing scale. If I put something that I know weighs 1 pound on that scale, I want my scale to tell me that this item weighs exactly 1 pound. There will be a bit of variability, of course, but in general, it will not tell me once it's 7 pounds and the next time it tells me it weighs nothing. So we want some metric that is measured reliably. And at the same time, we also want to minimize the risk of ambiguous decisions. And in practice, what that usually means is that we are focusing on one single metric to be the most important metric to be used in the study, because as soon as we use more than one, there's a risk that we get contradiction between the different outcomes that we think about. This single metric is usually called the primary endpoint, and that's the main focus that we use when setting up an experiment to decide whether or not a treatment is worthwhile investigating. Now, going back to Professor Moriarty's hypnosis therapy, there are obviously a wide range of possible endpoints, and usually there's going to be extensive discussions with the subject matter experts to decide what the most appropriate metric would be. But for example, whether or not somebody that has had SOA in the past is experiencing SOA after receiving the therapy would be one metric. It could be how often the sufferer has SOA after receiving the treatment, or we might be looking at how long does it take until the first SOA post-treatment occurs. Obviously, there are many other possible endpoints, and again, when deciding upon them, it is important to think about, first of all, what is meaningful to the patients, and in this case, I would hope all of these in some form or way are meaningful to an SOA sufferer. If I can show that I can reduce the number of sufferers that have SOA, that's a useful metric, I would have argued. If I can argue that the number of times a sufferer has SOA, that's going to be a useful metric to use. And similarly, if I can show that the intervals between onsets is longer, I have a useful metric to use. Leads me to my third question, and again, the question will appear in the featured section again, and that is one thing that I haven't thought about in my proposed endpoints, and for simplicity, I want you to think about the very first one, whether or not a previous sufferer does experience SOA after having received the therapy. And the question is, which time period would you wait to see if the hypnosis therapy does prevent SOA? So here we're thinking about, clearly, if we wait long, the chances of a recurrence is going to be higher. Are we going to wait for a year? Are we going to wait for half a year? Are we going to decide 1 month is enough? I can see some of the responses coming in, but a bit more hesitantly than previously. I'm just going to try to show you the results of this poll. So with about 10 responses in, you can see, hopefully you can see, that there are 7, or the majority of people have decided that 12 weeks is long enough. A couple are thinking, oh, I better wait for a full year, while there are a few that think 4 weeks is enough. Now, clearly, in this sort of hypothetical scenario that we are talking about here, and in light of the lack of information that I've given you otherwise, clearly there is no right answer to this question. But the important thing to think about is, what are the implications of that time period? If I'm thinking that SOA only happens to a sufferer anyway, very rarely, maybe it only happens once a year in the first place, then 4 weeks is probably going to be too short, because we will barely, just by doing nothing, we will barely ever see a sufferer have an SOA. At the same time, if this is a regular occurrence, then waiting for 52 months may just mean that everybody will have experienced an episode, no matter whether they had the therapy or not. And therefore, I will again not be able to distinguish whether or not my treatment is effective. Maybe the treatment reduces the number of episodes from 12 a year to only 1 a year. But if I'm looking over a 1 year period and just say whether or not they had one, then I will still not be able to distinguish and still not be able to claim that this treatment actually is helpful to patients, which just brings me full circle back to thinking about how to choose the primary endpoint in this context, making sure that it is meaningful to the patients, but also if there is a difference between taking the therapy and not taking the therapy, I want to be able to have to show that there is a difference in my experiment. Okay, so the next step then, once we've decided what our metric is, we're going to need to establish what it means to be better. And I'm stating here that outcomes of patients on the experimental treatment, in our instance, people getting the therapy, should be compared to people that did not get any intervention, untreated controls. And we want to make sure that these controls are sufficiently similar to the treated group so that if we find a difference between the two groups, it must be the treatment that makes this, that's causing this difference. And in order to establish that, in order to make sure that this is the case, one of the most useful techniques to use is randomization. Effectively, what it is we define who are the potential patients that take part in our study, in our experiment, and then we flip a coin. Are you going to get the treatment or are you not going to get the treatment? And because I'm flipping the coin, if there are any differences between the groups, they are also random. So if it turns out that somebody is male and gets the treatment and the next person is female, on average across the experiment, all these differences will sort of wash out, and therefore it will make our two groups as similar as possible so that the only difference is due to the treatment. A couple of other techniques that are useful in this context is to use a placebo control, and that means I'm going to use an intervention without an active ingredient. If it's a pill, that's usually just means I'm going to give a sugar pill rather than the pill that contains the actual medicine. Because it's been shown and it is well known that people that think they're being treated will do better than people that think they are not being treated. And I'll leave it to you to think about how on earth can we possibly do a placebo of a hypnosis therapy? How can we ensure that patients, participants in our experiments, do not know whether or not they got the hypnosis or something else? Another aspect that's useful in this context is blindness, and by that I mean making sure that both the participants as well as the assessors as well as the treating doctors, for example, do not know which treatment they are using. And again, the reason why we're worrying about this is because it has been shown that knowing the answer influences the outcome. And again, our overall objective is to make sure that any difference that we find must be due to the treatment. Now, going back to my randomization question, you might wonder, okay, so if I'm the physician, why can I not choose who goes in group 1 and who goes in group 2, who gets the therapy and who gets my placebo, doesn't get the treatment? To illustrate that, there's a little app that I would like you to try out. It's called How Random Are You? And the link is appearing in the featured section now. Effectively, what we're trying to do here is we're imagining we are a die, and we are rolling a virtual die in our head, and we want to decide what the outcome of that die is going to be. And so by going to that link, being a bit patient, waiting for the site to load, you get the instruction screen, and then you are asked, how random are you? And so you are asked to randomly pick a number from 1 to 6. And imagine you're being the dice, you're going to randomly roll a dice here. I'll give you a bit of time to make your choice, to pick. And while we do, what we expect, if we truly can imagine we are being dice that are randomly rolling a number between 1 and 6, what we would expect is that amongst all the people that are taking part in this exercise, on average, every number gets picked equally often. And so I'm just going to choose 3, for example. And you can see that there is now a bit of a histogram appearing. We need a few more responses for this to become meaningful. So I'm just going to try to reload. Let me just check that there are more people completing that little exercise now, because what we're imagining is that there is lots and lots of virtual rolls happening, each of us rolling 1 virtual die. Now, what you sort of start to see here, even on the basis of only 8 users, is that there seems to be a concentration in the middle. So 3, 4 and 5 have been chosen much more often than the extreme values. And if we do this with 200 people, what will happen is that comparatively very few people will choose 1 and 6, while the majority of people will choose 3 and 4, the middle numbers, just illustrating that we have a tendency to go into the middle with our guesses. And therefore, this is one of the reasons why we do not want to let the decisions about who goes into which group be made by the investigators, by the scientists, because no person can be truly random. Okay, so what I've done so far is I've just talked, based on a little toy example, about some good principles for how we can assess, how we can establish whether or not a treatment is useful. And now I'm going to start shifting focus on, so with that in mind, what's different during a pandemic? And in order to understand that better, I'm going to go back in time just a little bit. So what I'm showing you here is numbers of cases. So the 2 blue lines are different reported number of cases during an Ebola outbreak in 2014. So as you can see, at around March time, the first couple of cases have been identified, and then the number of cases increased until sort of November, December of that year. And then the outbreak seems to have died down. So in this case, we are in the sort of lucky situation where the epidemic itself controlled itself. It didn't go on exponentially, it didn't go any higher than about 500 cases a day before the trend reversed itself without any particular interventions. And I'm sure by now lots of you have seen the graphs of incidences of COVID going up and then coming down again because there's a lockdown. There are some interventions happening that change that behavior. Here we're looking at changing the behavior by itself. So the other thing to point out here on this graph are those vertical arrows, which corresponds to different clinical trials that have been started, that the time when the trial has been started and investigated potentially useful treatments. And if you look at that, I would argue that actually pretty much all the trials have missed the boat, right? I mean, by the time the trial started, by the time we have started to think about collecting information, in this case, the pandemic has already or is already dying down, and therefore the impact of any answers from such a trial is going to be minimal. Therefore, it is important that during a pandemic, a trial, any investigation, any experimentation for the new treatment must start rapidly to identify treatments. On the one hand, obviously, because this will help the patients, but at the same time, having an intervention available will also be part as a response to the outbreak. Now imagine that last year in April, we would have found a cure for COVID. Well, immediately the numbers would have dropped, right? And therefore, it is important to get things started quickly as possible. At the same time, however, there's a lot of uncertainties. It's a new disease. We don't know a whole lot about the disease at the onset, and so it is quite difficult to make choices like what is a meaningful outcome of my study? What's the right primary endpoint if I don't know anything about the disease at this point? And the final point to highlight is that frontline staff, frontline workers, who are the ones that are going to do the studies, that are going to give the treatments to patients, that are going to collect the data that we're going to use in order to make decisions about whether or not the treatment is useful. They are going to be under pressure already anyway. And so we're going to want to make sure that really we only collect information that is absolutely crucial so that we don't add to the burden on the frontline workers. Now with that, we get involved very early on in the outbreak of COVID. The so-called Lotus trial was a trial that started on the 18th of January 2020, so over a year ago now, and at a time where most people in the UK hadn't even heard of the coronavirus before. It's only 19 days after the very first reported case of COVID was available. Now, a consequence of that was that we had to make very radical decisions. So first of all, the treatment that we used is called lopinavir-ritonavir, which is a treatment that is currently being used to treat HIV. There simply wasn't time to develop a new treatment that we could potentially test in patients, and therefore the treatment that we have used is one that showed promise in lab experiments, but wasn't necessarily be developed for COVID specifically. We did, however, randomize in the study. We couldn't use placebo, unfortunately, because of the time it would have taken to produce a placebo treatment. The primary endpoint in this case was time to a 2-point change on this scale that is given here on the slide. And again, I'm not going to argue whether this is the most meaningful endpoint for patients. I'm not going to argue that this is the best endpoint to be used in such a study, but I'm going to tell you why we've chosen it. We've chosen this endpoint because it has proven to be a useful metric in influenza trials, in flu trials, and because we haven't had any experience with treating COVID patients, we used the best possible, if you want, alternative that was available. At this point, the thinking was COVID might be at least somewhat similar to the flu, and therefore maybe the metric used in flu might also be useful. After we got this trial started, we started to think very hard about what would the perfect trial during a pandemic actually look like. Now, a lot of the things I think are repeats of what I have already mentioned. They use meaningful and efficient endpoints. We minimize the burden on frontline staff because their main job is to save the patients. If they can do a study, an experiment alongside that, that's great, but they will always prioritize the safety and the well-being of the patients first, and therefore having a trial, a study that minimizes the burden is absolutely crucial. We also found that after having chosen one of the treatments that was potentially useful, that there actually is a fair number of existing treatments that might be useful to investigate. And so we were thinking it was important to include multiple treatments in a trial, and at the same time to be able to discard treatments that perform poorly, that basically have no hope of helping the patients as quickly as possible. And obviously the list goes on and on and on, but I'm not going to go into any more detail. I'm just going to show you with what we've come up with at the end of the day. I'm not going to claim that this is the perfect trial, but at least it has provided some answers that were desperately needed during the pandemic. And this is the recovery study, which started on the 19th of March, so not quite a year ago. You can see in my schematic that it compares 4 different treatments against standard of care, our control group. Roughly a month into the study, an additional treatment called tocilizumab was added to the study. Another month later, another treatment, convalescent plasma, was added to the treatment. And a week later, about 3 months after the study started, we had our first answer. We concluded that a treatment called hydroxychloroquine was no good for patients. Another 10 days later, dexamethasone was found to be useful in severe patients, and another 2 or so weeks later, we confirmed that lopinavir-ritonavir, the HIV drug, wasn't useful, and so on and so forth. Now the trial is still ongoing, and therefore, but I ran out of space on my little schematic here, but I hope you see the picture. We started the trial quickly. We've made it such that we can find decisions, we can make decisions about potential treatments as quickly as possible, both because they're good and because they are not helpful. We've tried to minimize the burden on the healthcare staff, which meant that the healthcare staff, the frontline staff, was able to deliver and find patients to take part in the study. Now, because our pandemic, unfortunately, is still ongoing and we're still in the midst of it, I've decided not to finish up with any sort of conclusions, because I'm afraid the current pandemic hasn't concluded yet. But I will stop here and ask if there are any further questions. If you see me looking to the side, I'm just looking at the featured tab to make sure I don't miss any questions that might be posted on the Q&A.
Thank you very much. Can I just come in with a question? So have you noticed a difference in how clinical trials have been carried out during COVID compared to pre-COVID?
Yes, there's definitely a couple of things that are notably different. So the first thing is that COVID trials tend to be much more streamlined in the kind of information that they are collecting. They're much more streamlined in terms of all the documentation and everything else that goes around with conducting a clinical trial. And I've sort of alluded to that we want to minimize the burden on everybody involved, and that I think has been taken on across the board with most clinical trials that look at COVID treatments. Collect only information on the things that you truly think you need, because traditionally there is a bit of a sort of thinking that, you know, I'd rather collect the information in case I might need it later. And so a lot of traditional trials will collect 200, 300, 400 different outcome variables because they might prove useful, while within COVID, the decisions are reduced as much as possible, collect maybe 4 or 5 different metrics that you might be interested in, maybe 20, but not a couple of hundred. The other thing that has definitely changed is that there's a broader use of so-called adaptive methods and so-called trial platforms, which effectively just means you have the infrastructure in place to run an investigation, and that infrastructure stays in place. And if there's a new question, a new treatment that appears, gets into the mix, it will be added to the study. And so recovery, for example, is one of these platform trials, one of these platform studies that have been used very rarely before COVID, but now are very widely used as part of the sort of response to COVID.
Excellent, thank you. So we do have a few questions coming in. So firstly, there's a question: how much have your skills developed during this pandemic?
It's a good question. They have definitely, the skill set required has definitely changed, in the sense that, again, the sort of trials designs that are being used and are probably most useful during a pandemic are different, and therefore it's obviously necessary to adapt and to learn about these new methods and do investigation, do research to make sure that these new approaches that you are using are appropriate. And so there's definitely a lot of learning gone in to make sure that, you know, you set up the most relevant trial for the most relevant question.
Okay, and then another question: do you think that the pandemic has made researchers think more carefully and analytically about how they are conducting clinical trials?
I think the answer to that is a yes and a no. There's definitely some very careful thinking about streamlining, making sure that you can run your study efficiently. There's been a lot of thinking and sort of improvements done in that respect. I mean, just to give you a bit of context, it's not unusual for a traditional study to take 6, 9, 12 months to even get started because of the paperwork required, contracts and so on and so forth. And a lot of effort has gone into making sure that all these sometimes perceived barriers to moving quicker are reduced or removed completely. So there's been a lot that has happened there. At the same time, because of the urgency, I think there is still a lot of new work to be done to think about. So what would have been the best way to design a clinical trial during a pandemic? You know, the solutions that we have put out there have proven useful, but I think once things settle down, and I'm confident they will, we're going to have to look very careful at what has happened and what has been done, and think about whether we could have done it even better next time around.
Okay, and so we do have a question that's come in about the AstraZeneca vaccine and the blood clotting. As a statistician, what would your approach be to understanding the current concern regarding the AstraZeneca vaccine?
Yeah, it's obviously a question that many people is on many people's minds. I think it is important to realize that safety monitoring for all interventions, vaccines and otherwise, is very rigorous. And this is exactly what we are seeing here with the suspension in some countries of the AstraZeneca vaccine. Now, my interpretation of what I have seen is that there is no strong evidence that there is an increased risk of harm from the vaccine. Now, if you look at the sort of information that's available and so on and so forth, depending on how you slice and dice and distinguish the different types of clots, your answers will vary a little bit. But overall, my impression would be that the risk of harm, if there is any, is very small, and it definitely is outweighed by the benefits of the vaccine. And so I personally would not hesitate for a second to receive the vaccine at this point.
Okay, we have had just a comment about the sound. Hopefully everyone can hear both of us. So another question, was proning included as part of the trial?
I'm not sure exactly what's meant by proning in this context, sorry.
Okay, shall we move on? We've got another question. How do the research scientists...
Maybe whoever asked that question could clarify what exactly they mean by proning.
Yeah.
But in the meantime, I think we can definitely look at the next question till we get confirmation.
Okay, excellent. So how do the research scientists know which medication to try in a pandemic? For example, I'm afraid on my pronunciation of that, but hopefully you can see that Thomas is currently used to treat. Yeah, so if you can take that from there, thank you.
Yeah, that's a very good question. So there are a couple of different sort of studies and investigations that tend to happen before treatments actually get used in patients and trialed within a trial. So what typically happens in the first place is to identify whether or not, if it's a virologic agent, for example, whether or not that treatment is potentially useful to kill that particular virus. And so on the basis of lab experiments, the antiviral agents were screened against checking essentially whether or not they, at least in the lab conditions, have potency against the virus. In the case for tocilizumab, which is an anti-inflammatory agent, essentially the rationale was based on the, at that point, better understanding of the disease trajectory and the fact that patients with severe disease would come into a hyperinflammatory state. Now, I'm a statistician, so I'm afraid that's about as much as I know about this aspect. But there's a condition called hyperinflammatory state that it was thought and understood that interventions such as tocilizumab could modify and reduce that inflammatory state. How exactly tocilizumab was chosen rather than any of the other biologic agents that are available, my understanding is that, at least for the recovery study, one of the aspects was the treatment needed to be available. It needed to be available in large numbers, large enough numbers to actually make a difference to the patients. It's no good if I test a treatment where, after I've done my study, I can treat 10 other patients because I don't have enough of the product anymore. For tocilizumab, it was understood that there were at least sufficient doses available to be used afterwards if it was proven successful. In addition to that, of course, tocilizumab has been shown to be useful in arthritis, and on that basis, that decision was made. So the next question that I can see here is whether the use of CPAP was included in the trial, and I suspect that that question is sort of linked to the question of proning. So if I'm I haven't seen a clarification of that question, but I'm assuming we're talking about whether the patient is being put in prone position in order to alleviate breathing troubles. Basically, this is part of standard of care, and so in the trial, the control group would get whatever the best current understanding of treatment is, and if that includes putting the patient in prone position, if that includes putting the patient on CPAP, they would be put on those interventions. At the same time, everybody that gets an active compound, everybody that gets an experimental treatment, will also get exactly the same standard of care, plus on top of that, whatever experimental agent is used, and they have been randomized to.
Brilliant. And then just final question that's come through. Has there been a good degree of global uniformity in the architecture of such COVID trials across the world?
Again, it's a bit of a mixed boat, if you want. There has been great interaction on some levels. On the other hand, of course, other activities have unfortunately been duplicating. So what do I mean by that? The WHO, for example, has set up a very large study that's run in multiple countries across multiple different treatments, for example, with a view to collect information across different regions so that information is collected more quickly. There's been great interaction between the recovery group and the WHO group. There's been great interactions with the US Institute for Infectious Diseases, for example, on their studies. So there's been a lot of conversations ongoing, and so that's been definitely useful and been very positive. At the same time, of course, there still has been some duplication. So I've just in my two examples that I've spoken about, lopinavir ritonavir was tested in China initially, and then again included in recovery because the information wasn't shared, wasn't strong enough to make a definitive conclusion. Several other treatments that are being investigated in multiple studies, and even though replication of results is useful, I don't think you need 100 studies that all look at hydroxychloroquine at the same time, for example. It would have been much better to collaborate on these. And so I think there is, again, some learning for the future in how can we better work together and get answers quicker rather than duplicating effort, although some of that, as I said, has been avoided, and at least information sharing between the different groups has been pretty good.
Okay, great. So we just had one, I think the person that was asking specific to the proning, just with a follow-up question, the recovery trial was a trial of pharmaceuticals only, is that right?
That's correct, yes. So the actual interventions are pharmaceutical interventions only. So we did not look into whether positioning the patients differently improves or changes their outcome. As I said, if it was felt to be in the best interest of the patient to be put in that proning position, they would be as part of the standard of care, as with a view of we're not withholding anything that seemed to be useful for that particular patient, but then they would be on top of that would receive an experimental pharmaceutical agent if they're randomized to it.
Okay, excellent. Do we have any more questions before we close the event? Okay, I think that's all our questions then. Thank you very much. Oh, sorry, just. And thank you very much for your feedback, and it's great to see that you've enjoyed the session and found it interesting. Thank you very much to Thomas for speaking, and thank you all for joining us today. It's been our first virtual Science Festival event, so thank you very much for coming along, and take care and have a good day.
Bye.